Welcome to the faculty talk. We have we have done this for some time back, and we are restarting this faculty talk. I think about 3-4 years back, the faculty talk was a running event in the Faculty of Agriculture and Plantation Management. So we try to inculcate research in the faculty by organizing such events, and we hope to start this from this year. Again, restart this faculty talk from this year. And for the first faculty talk, we have Dr. Janaka Wickramasinghe, who has conducted his PhD in Iowa State University in USA. So this is part of his PhD work. So the importance of faculty talk is that we try to get the new things that people learn from going abroad and get it disseminated in ourselves. Right? So we try to understand, we try to learn from what they have learned, what they have done rigorous research in their PhD time. And although we start today, we hope to bring in more students into this. I will make it compulsory for all the research students to come in from the next faculty talk, so that this will be popular among students. So students should see what the new things happening in the faculty, and we plan to have outside resource persons as well. And we will bring in from Peradeniya University, we will bring in from other faculties in our university, and we will bring in from Kalaniya, Jayawardhanapur, everywhere, and get whatever the new things happening in the research world into the Bible University of Sri Lanka. So I thank for Dr. Janaka for agreeing in such a short notice. I asked him in 2 days back whether you can start this, and he kindly agreed. So that is why we have less of a crowd, because we could not advertise this as much as possible. But Dr. Janaka kindly agreed to do this in a very, very short time, only 2 days. And let's hear from him. Thank you very much.
Thank you, sir. Yes, as Jagat sir said, I was informed about this presentation by last Sunday around 7 p.m. So then I agreed, okay, we will do somehow. So then actually we can continue in the future months as well. So fortunately, I said to sir, so I will be conducting one of my research in the my PhD degree and my master's. So together, so I will be kind of selected one specific research that I conducted, and then I will be kind of talking about that. So basically, my talk would be basically on nutritional interventions on dairy calves, especially who are undergoing stresses, right? So before that, I think most of you maybe know about me, but just to give you some kind of background about me. So I joined Faculty of Agriculture and Plantation Management in 2014 as a kind of a probation lecturer under the headship of Professor Janaka Wickramasinghe, and Dean was Professor Attanayaka. So then as the Vice Chancellor, I think Professor Jayasekara. So he was there and the introvert, and fortunately Chandan sir is here. So he was my first and former head of the department. And then I confirmed as a lecturer in 2019 and promoted to the senior lecturer, grade 2 in 2019. So my educational background, I did my undergraduate degree in University of Peradeniya Agriculture Faculty. So I completed my agriculture technology and management degree in specialization in animal science in 2012. And then I joined Wimber University, as I mentioned, in 2014, and then I went for my master's or the postgraduate degree in 2017, and completed both my master's and PhD in Iowa State University in USA, and came back to Sri Lanka in 2022 July. So that's kind of a brief introduction about myself and my professional background and my educational background. So when I kind of started my journey as a postgraduate degree kind of a seeking student in Iowa State University, my main area was focusing on dairy calves, specifically dairy calf nutrition. Since there are some students, maybe sometimes I'll be talking in Sinhala as well, just to kind of explain some aspects as well. So basically, I was focusing on some aspects of dairy calves, basically related to the USA or kind of a Midwest area, because my university was kind of located in the mid of United States of America, which is very close to the Canada and the other kind of major states like Wisconsin, Nebraska, and those kind of states. So those are basically identified as major dairy performing states in the USA. And therefore, when kind of went through some literature, we found that most of the cows, like the young ones or the mature ones, are basically kind of replaced. About 34% of mature ones are kind of permanently removed every year from a particular batch. එක අපි සාමාන්‍ය ෆාම් එකක් ගත්තාම මේ ෆාම් එකේ 134ක් විතර ඉන්න mature cows ලා අනිවාර්යෙන් අවුරුද්දකට සැරයක් replace කරන්න ඕනේ. ඒ කියන්නේ අපිට හැමතිස්සෙම මේගොල්ලන්ව ගන්න වෙනවා අලුත් කට්ටියකින් replace. අපි දැන් සාමාන්‍ය යුනිවර්සිටි එකක batch එකක් යනවා වගේ අවුරුද්දකට සැරයක් අනිවාර්යෙන් එක batch එකක් අයින් වෙනවා. Right? So the main thing is the raising of dairy calves, right? Which is very expensive. So in terms of, I can show you some kind of data. So if you kind of see kind of the cost associated with dairy calf management, right? So average cost per day, this is in 2019 and 2020. The values, now the values are kind of comparatively high because of the cost of living and the inflation. Even in USA, they have the same experience like we have. So the average cost is about $6 per day. ඒ කියන්නේ සක්ක දවසකට එක පැටියෙක් රෙයා කරන්න $6ක වියදමක් දරන්න ඕනේ එක ෆාම් එකකට. දවසකට $6ක්. Right? In Sri Lankan rupees, which is about almost 2,000 rupees a day. Right? අපි 300 කියලා හිතුවොත් මේක ඩොලර් රේට් එකක් යන රේට්. Right? So however, if we can wean them, right? If we can stop giving milk to those animals, right? At least as much as earlier, right? So then it will be kind of going down maybe at least 50%, which means on average about $2.5 per day can be kind of spent if we wean from milk these animals, right? So basically the cost is associated with the whatever the milk cost plus the labor cost, right? Because the management of these animals are kind of very expensive because we have to give individual attention to these animals, not like the large or the mature animals. So therefore we can reduce the cost at least by 50% if we can wean them as much as possible, right? So that's first basic kind of idea that we had. However, still, even in USA, the average weaning age, අපි සාමාන්‍ය සිංහලෙන් කියන්නේ කිරි වරණ වයස කියලා. අපි කිරි දෙන එක නවත්තන age එක, right? So basically it is about average 9 weeks. So we have to wait until maybe about more than 2.5 months just to kind of stop giving milk to these animals, right? However, if you kind of go through different literature, we can find that on average we can wean them or we can stop providing milk for these animals maybe once they kind of reach 6 weeks of age, right? අපි කොච්චර කිව්වත් ඒගොල්ලෝ උනත් සාමාන්‍යයෙන් තාම සති 9 10 12 වෙනකන් යනවා රිසර්ච් වල කොච්චර පෙන්නලා තිබ්බත්, right? So if you kind of calculate the cost, right? So this is the difference, right? If we go for about 63 days or 9 weeks, so we have to spend about $378 just to kind of complete that particular time only for one particular cow. එක සතෙක් වෙනුවෙන් මෙච්චර වියදමක් දරන්න ඕනේ සති 9ක් ඒ සතාව අපි manage කරනවා. If we are going to manage until kind of 9 weeks, so we have to spend this much. So just imagine when they have about 2,000, 3,000 young cows in one particular farm, right? Even in the farm that I conducted my research, our university farm in Iowa State, so we had about 50 to 60 on average dairy calves which are kind of under milk management. So අපි සාමාන්‍යයෙන් 60ක් විතර manage කරනවා. So just imagine the overall cost associated even in a kind of a small scale farm, right? So if we can wean them at least about 6 weeks, right? And then continue up to 63 days with post weaning it, still it is about $305. Even it's still less than that one, which means still they can save about $70 from one particular animal. So which is kind of a huge benefit if we can cut down this age to 6 weeks, and we need to kind of justify. It's like if you kind of consider this age in Sri Lanka, which is about 4 to 6 months. සාමාන්‍ය ලංකාවේ NLD farm එක ගත්තොත් මේක සාමාන්‍ය සති 12 ඉඳලා 16 වෙනකන් යනවා. Right? So you can imagine why they are kind of losing some money, because we are spending lots of money on milk feeding for these animals. So that's the basic condition or the concern we had when I went through the kind of the literature review before I start my kind of a postgraduate research studies, right? So therefore what we have to do is we have to enhance their growth, right? The main target just to kind of enhance the growth. Then we can kind of cut down whatever the amount of milk that we are going to offer for these animals, right? So when we kind of cut down milk, the major concern is it can adversely affect on their immune system and the digestive system, right? So those two are the main things that can be adversely affected, right? So in those two, I was basically targeting on the digestive system or the nutritional factors affecting on their digestion or the digestive system of that kind of animal. So that kind of a things can be maintained or what we call the managed through different factors like colostrum feeding. We know that even the humans, we kind of drink lots of colostrum at the initial stage of our life. Then the seasonal variation, housing and bedding. So if we can manage those aspects, right? And also the diet and nutrition. So those are the main factors that we can kind of control just to kind of support proper growth and development of these animals. So this is just to show you some kind of image, right? So if the animal is giving only milk, I think most of you have done agriculture, you know this one, at least you have some basic background in animal science. So this is the rumen, අපි කියන්නේ අර romantic සත්තු කියලා rumen එකේ තියෙන developments, right? So you can see that the rumen is not well developed if they are getting only milk. So there are some arguments in the society or maybe some people who are kind of against animal science arguing that why we are going to cut down milk for these animals because that's the right of these animals, right? අපි ගොඩක් දැකලා තියෙනවා ඔය වගේ ප්‍රශ්න. Even in USA we had that issue when we were kind of performing this kind of research. There are some animal activities, they are kind of protesting again this kind of an initiative, just cutting down of milk from 9 to 6 weeks or something, right? Because they are thinking, okay, this is their right. But these animals are born to be ruminants. That's the main thing. අපි කොච්චර මේවා කරත් මේ සත්තු ruminants ලා. We cannot change their physiology, right? So therefore, we need to kind of provide some support just to develop their rumen and be kind of an independent animal from milk. So that's the main target, right? Even though we say so, අපි කියන්නේ අර පව් මේ සත්තුන් කිරි දෙන එක අම්මගෙන් අයින් කරනවා එහෙම කියලා එහෙම කොච්චර කිව්වත් මේ සත්තුන් රිසර්ච් තියෙනවා. පුළුවන් නම් බලලා ඔයාලා බලන්න පුළුවන්. So they have done some research just giving milk for maybe 6 months, 1 year, maybe 2 years, only milk, but they will not be survived. So that's the case. So because they need to kind of be a ruminant, they need to eat some grasses or any kind of forages, rough materials, otherwise these animals cannot survive. That's the kind of a basic kind of a nature that we have to understand or accept, right? So, however, when we kind of initiate this kind of a milk cut down method, we usually call it as weaning, right? Or kiri waranawa කියලා අපි කියන සිංහලෙන්, right? So the main concern is when we are offering some milk, the abrupt cutting down of milk can adversely affect on these animals, right? අපි ඕනම දෙයක් දෙනකොට අපි අපේ ළමයිට උනත් අපේ අම්මලා කිරි දෙනකොට උනත් එකපාර කිරි දෙන එක නවත්තන්නේ නැහැ. Just we try to kind of give them some chance to kind of consume some or use other hard or whatever the dry matter or any other kind of external matters, and then slowly, gradually cutting down the milk. So these two graphs just to show you that matter, right? So what happens if we kind of gradually cut down the milk or if we kind of cut down milk abruptly, right? එකපාරට කිරි දෙන එක නැවැත්වීම සහ කොටස් වශයෙන් කිරි නැවැත්තුවාම මොකක්ද වෙන්න පුළුවන් පොඩි පොඩි වෙනස්කම් කියන එක, right? So this is just to kind of show you that. So if we kind of giving or cutting down milk slowly, what happens is the animals will gradually catch up whatever the dry matter from the concentrate or any other forages or any other grasses available surrounding from their environment while drinking some milk. So that's the main kind of a concern about these two graphs. So that's what I wanted to kind of highlight, right? So this is just to show you that too. So what would happen if we cut down the milk gradually and abruptly cutting down milk? So the rumen development can be kind of enhanced or initiated if we are cutting down milk gradually, right? Similarly, the small intestine development and everything kind of can be enhanced if we are going to cut down the milk at gradual manner, not like abrupt cutting down of the milk, right? When we kind of cutting down milk, the main thing is it will adversely affect on their intestine, the small, mainly small intestine. So you can see that our small intestine is kind of arranged in kind of a manner that with most of the intestinal epithelial cells. අපි අර හැම උඩ තියෙන අපේ හැමේ තියෙන cells වගේම තමයි epithelium එකක් තමයි මෙතන තියෙන්නේ. So which are kind of connected with the tight junctions, or the proteins which are called tight junctions. So I think if you can remember in some kind of anatomical features of the digestive systems of the animals, right? So they are basically connected in these tight junctions. So as they are born, they are not kind of tightly kind of bound to each other. So that's why we say, yes, colostrum feeding is essential. So everything can be absorbed via these junctions, right? අපි අර මුලදී ඉපදිච්ච ගාම එක colostrum feeding කියන එක වැදගත් කියලා කියන්නේ ඔය tight junction හැදිලා නැහැ ඉපදිච්ච ගාම සත්තුන්ගේ. සත්තු ඉපදිලා ටික 24ක් උපරිම අතනද මේ tight junction bound වෙලා absorption එක නවතිනවා, right? So then we have the selective absorption. Until that, we can have kind of a complete or kind of without any control, everything will be absorbed into our body. So that's why colostrum feeding is kind of essential. This is kind of an additional information, but that's why the same thing would happen if something happened to our digestive system even after maybe about 5-6 weeks of time, which can be initiated through the weaning or cutting down of nutrients of these animals. What happens is this tight junction can be damaged. With the damage of that tight junction, what happens? These whatever the nutrients plus pathogens, toxins, and everything can be absorbed into the body of these animals. So eventually it will kind of affecting on their immunity as well, right? So that is called basically the paracellular permeability. So even we as humans, I think we are doing that test even in some cases when we have some issues in our digestive system or the small intestine. We do this kind of a test, we call this paracellular kind of permeability assessing test. We use some glucose or maybe mannitol or something kind of a different sugar molecules or markers just to assess the paracellular permeability of these animals. This is just to kind of give you some idea what are the main tight junction of protein. We have the chlorine, octodine, and soluble octodine. So there are different types of tight junction proteins available. So these things can be damaged. Once they are damaged, what happens? These two cells, there will be kind of an opening, which is kind of a free for any organism to enter into our body without any control. So one of the issues we had in USA, they were feeding lots of milk for these animals, which means per day they were feeding about 6 liters minimum. In our farm, we were feeding about 9 to 10 liters of milk per day for these young animals, which means very high volume. Just to compare, imagine what is the volume that we are feeding. In Sri Lanka, we are feeding only 3 liters per day for these animals. So අපේ ලංකාවේ මේකවල පීඩ් කරන ඉපදිලා ඉන්න පොඩි සත්තුන සති 12 වෙනකන් දවසට කිලෝමීටර් 3යි average. NLB farms වල data නම් බලන්න පුළුවන්. මේගොල්ලන්ට average 6ක් feed කරනවා. අපේ හිටපු farm එකේ now minimum feed කරනවා per day, 3 times per day. So they are growing very fast because there are lots of research showing that if you feed or if you provide more nutrients freely in age, අපි කිරි වරන් ඉස්සලා වැඩිපුර nutrient දෙනවා නම් what happens is they will be growing really fast. Or they will achieve their puberty and those kind of things very soon compared to the animals who are providing less volume of milk. However, the basic disadvantage is if you are providing very high volume of milk, ultimately their grain intake or whatever the concentrate or the dry matter intake will be going down. So that's the main thing. But what we want is we want to enhance whatever the dry matter or concentrate or dry matter intake to their best or the optimum level. So that's what we want to do. So therefore, that's why we wanted to kind of conduct a research in kind of a step down meaning manner, right? The other factor I was kind of thinking, there were two major issues we had during these stresses. Other one is the heat stress or the heat waves. So where I was kind of doing my PhD is kind of a state. It had kind of a 4 seasons, clear 4 seasons, and in the summer or the summer season, we had kind of a day temperature which is almost over 32 Celsius degrees. අපි දැන් ලංකාවේ නම් මේක එච්චර හිතන ගන්න නැහැ. මෙහෙ වුණත් 35 විතර කියලා හිතන්න පුළුවන්. But if you go to a country like USA or maybe close to the Canada, which means this is kind of a stress temperature for anybody who are living there. Because only average temperature is about 10 to 15 Celsius degrees in a normal day, right? But if you kind of think about this, which means almost 100% or more than double the temperature they have on average on other season except for the summer season, right? So then this was another factor affecting on the kind of a health of these animals. So then I was kind of focusing on this factor as well, as together with the weaning plus the heat stress for these animals. So what could happen when the animals kind of are suffering from some heat stress is eventually it will adversely affect on their digestive system. Like weaning, what happens is their matter intake will be going down, and it will eventually create kind of a psychological or maybe oxidative stress to their digestive system and eventually damaging their intestinal tight junctions. So then it will open again and kind of expose to the outside environment, which means eventually the toxins or the pathogens available within the body or coming through our digestive system, right? So those two were the kind of main factors I was highlighting or kind of targeting or literature searching during my PhD or at the beginning of my PhD, just to kind of find some solutions. So because there are lots of research have been done with this swine, right, as well as the mature ones or the adult cattle, but unfortunately there are a very minimum of research have been conducted regarding the dairy cattle young animals or growing heifers. අපි කියන නෑම්බියෝ කියලා කියන්නේ grow වෙන සත්තු. wean කරපු නැති නැත්නම් කිරි වල්පු නැති සත්තු plus ඒගොල්ලන්ගේ ඊළඟ stage එක කිරි දෙන්න ඉස්සෙල්ලා. පැටව හම්බෙන්න ඉස්සෙල්ලා අපි නෑම්බියෝ කියලා සිංහලෙන් පාවිච්චි කරනවා. So those two stresses I was kind of basically focusing on during my PhD. So if we can kind of support these two stages, at least to some extent, what we have done is we can kind of increase their growth. At least we can minimize whatever the growth slumps or maybe the dry matter intakes, whatever the slumps we can see because of these two stresses. So if we can manage or if we can mitigate, we cannot kind of eliminate 100%. No, nobody can do that. But if we can kind of control that one to some extent, we can get some benefits, and in also it will enhance their welfare aspects as well. So we were kind of targeting about two nutritional interventions for these animals. One is functional amino acids, the other one is phytogenic feed additives. I think this one is, I think, very familiar with our faculty as well, because I think Chandana is also here. So he is also doing some research regarding antioxidants and some plant origin materials. So we were kind of targeting on phytogenic feed additives as well as functional amino acids for these animals and try to kind of mitigate or control these stresses of heats as well as weaning, right? So as amino acids, I was kind of basically targeting on some non-essential amino acids. Of that, main target was given to glutamine. Glutamine means one of the non-essential amino acids, but the thing is, during stresses, it usually become essential, which means kind of a conditional essential. They can make a සාමාන්‍යයෙන් අපේ ඇඟ ඇතුලේ හැදෙනවා. ඕනම සතෙක්ගේ කිසි කන්ට්‍රෝල් එකක් නැතුව හැදෙනවා. නමුත් අපි ස්ට්‍රෙස් එකේ ඉන්නකොට මොකද වෙන්නේ? මේක conditionally essential වෙනවා. ඒක automatically essential part convert වෙනවා. So therefore I was targeting on this amino acid that is called glutamine. The other one is branch chain amino acids. There are three branch chain amino acids which call isoleucine, leucine and valine. So these are essential amino acids. So we cannot produce these things our body. We can, not to the adequate amount. So anyway, we have to take them outside of the body, which means from the feeds or whatever the supplementation. So that's why we have to kind of consider them as essential amino acids. So this is just to show you what are the basic functions done by these amino acids. So with the time, so I will just skip those things. So there were some research have conducted regarding glutamine and branch chain amino acids on the swine as well as some other livestock species, but not for the dairy calves. So therefore, I selected this supplementation and branch chain amino acid supplementation for one of my research during my PhD period. And because this was the baseline, there was no any other information available. Only the baseline was given by this research that we found from the swine. මේගොල්ලන්ගේ තිබ්බ data විතරයි අපි ළඟ තිබ්බේ වෙන කාලවලත් කරලා තිබ්බේ නැහැ. ඒ data ඔක්කොම use කරලා තමයි අපි baseline එක හදාගත්තේ. මේකේ කරොත් මෙහෙම වෙයි කියලා hypothesis එකක් හදාගෙන අපි ඉස්සරහට ගියා glutamine and branch chain amino acid supplementation එකත් එක්ක. Right? On the other hand, we had phytogenic feed additives. So as we know, so there are lots of benefits which are basically bioactive compounds extracted from plant materials or the metabolites, right? So antimicrobial, antioxidant, anti-inflammatory effects. There are lots of effects have been found or recorded, or if you go through the literature, you can find those things very easily. So the main thing is they are kind of a supporting the inflammation. අපි ඇඟේ ලෙඩක් හැදුනොත් හරි මොකක් කරනොත් ඒක mitigate කරන්න control කරන්න තමයි ගොඩක් වෙලාවට මේ fiber feed additives වලින් support කරන්නේ. I think this is very familiar words for you, the vanilla, licorice, caraway. So we were kind of targeting or looking for kind of a product which already have in the market in the USA. So using for this particular kind of a research. So then we found kind of a product developed by a company which called Biomine company. So they were agreed to kind of test their product on dairy cows as well, because this was kind of found to be given some support with dairy cows as well as some beef cattle. Right? Beef cattle කියලා අපි මම හිතනවා ගන්න නැති කියලා beef cattle කියන අපි මසට. We do not have kind of an industry in our country, but in other countries they have different types of animals called beef cattle, or they are called steers. So they are basically only for meat production, not for any kind of a milk or whatever. So they are targeting on meat production only. So they have been done some research on the mature ones, but not for the growing animals. So therefore, they agreed to provide their product and provide some funds to just to conduct some research for testing this product even under the heat stress, because they have found some kind of a support of that product to kind of mitigate whatever the heat stress for these young animals, right? So my overall objective of my PhD thesis was just to kind of try to kind of find information on the effect of functional amino acids as well as phytogenic feed additives on young cows who are undergoing weaning as well as heat stress kind of a conditions. So there were some specific objectives. So I conducted 4 different research, including laboratory research, because I will not be explaining those things, but we did some field research, and then just to kind of understand the molecular mechanisms, we tried to kind of isolate initially some intestinal epithelial cells and cell culture, but unfortunately we were not successful at that field. So then we were trying to find out separate cell line. Dr. Priyamada හැමෝටම දන්නවා අපි cell lines වෙන වෙනම තියෙනවා available. Cancer study කරන්න එහෙම නැත්නම් අපිට molecular level එකේ study කරන්න අවශ්‍ය නම්. So we obtained some cell lines from other universities, and even there were some cell lines in our other labs as well. So we try to kind of mimic whatever the things that we observe in the field level, in the laboratory level as well. So that's what we did, just to understand the molecular mechanisms, so what we saw in the field experiment. So there were 4 specific areas that I was targeting during my PhD. So one of them was kind of in vitro study, just to kind of understand the molecular mechanism, and all the others were field research. So today I will be just kind of a brief what I did with the dairy cows when they have kind of a heat stress with the phytogenic feed additive supplementation. So our hypothesis was just to kind of providing the feed additive, the phytogenic feed additive, which is basically that I mentioned earlier, the digestron product, on heat stress animals, just to kind of mitigate their heat stress and just to kind of support to kind of avoid any inflammation or growth retardation. So that's what we were kind of trying to do during this particular research. So for this, we used growing heifers. There were 22 dairy cows. So under this kind of a setup, so we selected the 22 heifers, and there were 2 basic treatment groups. One is the control. They were given only the basic diet, that is called TMR diet, total mixed ration. So the next one we were giving the TMR plus the supplementation of the product or the phytogenic feed additive as per their body weight basis. So it was about 0.25 grams per head per animal per day. So that's what we kind of did, and this was ran for about 25 days. So that's it. අපි මේ research check කරා මේ farm එකක් අපේ farm එකේ. So this is the experimental timeline. So what we did, there were about, before starting any kind of animal research, one of the major law in USA is we have to kind of provide them kind of an adaptation period. So minimum adaptation period is about 7 days. So otherwise we cannot just expose them to any kind of treatments. So we kind of supplied them about 7 days of adaptation, and then we had to kind of collect the baseline data without exposing to any treatments, and then there are 7 days of heat stress period. දවස් 4 heat stress එකකට අපි expose කරා මම ඒක කියන්න කොහොමද අපි කරේ කියලා. So after that then we were kind of focusing on what happens after the heat stress as well, just to kind of see whether they are kind of catching up to the level that we had during the baseline, or at least just above the whatever the baseline data. So that's what we were doing, and we kind of collected their body weights, and we collected their bloods as well as their vitals. Vitals means basically their physiological parameters such as rectal temperature, skin temperature, heart rate, heart the movements, and everything were recorded. Even we scanned animal with the kind of a scanner just to kind of see the blood flow to this skin and all those areas of the animal, just to see whether any blood flow movements based on the heat stress that we are going to provide for these animals. So this research was conducted at the Dairy Research and Teaching Unit at Iowa State University. So we had a farm with about 600 dairy cows, milking herd. So we did all the kind of research over there. So this is kind of a metabolic barn. So they had kind of partition cages, and then we just kind of divided those animals into different treatment groups, and then we just allocated whatever the waterers and feeders as per their requirements. And all the animals were kind of provided with all the required bedding and everything, because these are kind of given as a law. They usually come and time to time check, as we discussed about the research ethics committee. So they have a different committee. So time to time they come and see whether we are doing the research properly or not. So if we are doing any violations, so they will just stop the research, and then they will prohibit whatever the professor or the group to conduct research for a particular time period. So otherwise we have to kind of adhere whatever the guidelines that they are given, and approval and follow whatever the ethics and everything they have imposed to conduct the research. So based on their requirements, and we try to kind of adhere all the guidelines, and then these were some data collections what we did. So we had to kind of collect the blood samples time to time, and we were kind of testing their what we call digestive permeability. So because just to kind of see whether these animals are damaged with their digestive system or the intestine, we can do two things. The one thing is we can do this kind of a method. We can use chromium EDTA plus some sugar molecules, and then give that animal to drink, and then collect the urine samples or blood samples, and do the assessment in the laboratory level. Or what we can do is we can kill the animal, get the tissue samples, and conduct the animal. So, but if you are going to pay this kind of kill means sacrifice this kind of animal, we have to pay about $500 to $1,000 based on the stage of these animals. So therefore we did not kind of initially started such kind of a research. We just use these kind of external molecular markers just to see whether any kind of damages has been done for these animals. I don't know. Yeah, this is the setup. I don't know whether it is, yeah, you can see. So these animals were kind of separated into different units and different treatment allocations. So completely closed unit environment. So then we turn on the heaters. The heaters were on the roof, and then we just turn on the heaters for about 12 hours per day, and then the remaining 12 hours we just stop kind of providing the heat. So මේක අපි ගොඩක් වෙලාවට මෙතන හීටර්ස් තමයි උඩ තියෙන්නේ වහල හයි කරලා මේ කරන්නේ පෑ 12 උදේ 6ට ඔන් කරනවා. උදේ 9ට ඔන් කරාම රෑ 9 වෙනකන් එක දිගට හීටර්ස් දාලා තියෙනවා. ඒතර ගොඩක් වෙලාවට මේගොල්ලෝ හී ස්ට්‍රෙස් එකේ ඉන්නවා පෑ 12. ඉතුරු පෑ 12 අපි කරන්නේ heaters off කරලා ඒකලා normally ඉන්න දෙනවා. So this is how we observe what you see when they are under heat stress. මේ වෙලාවට තමයි මෙහෙම සත්තු කන්නේ බොන්නේ නෑ වැඩිය. මේගොල්ලෝ නිකන් අර just stress අපිට ඒක බැලුවාම තේරෙනවා ඒක. අපිට උනත් ඔක ඇතුළේ ඉන්න බෑ. මම උනත් විනාඩි 15ක් 20ක් ඉඳලා එළියට ගිහිල්ලා යතුල්ට එනවා. මොකද අපින්නත් ඉන්න අමාරුයි ඔක ඇතුලේ. they are just kind of shivering, just like shivering. They just try to kind of dissipate their heat as much as possible. That's what they were trying to do. So no kind of eating any feed, just drinking water. That's what they are usually do under the heat stress. So this is one of the observation you can see once they are heat stressed. So they just maybe in the corner and just try to kind of dissipate the heat. Salivation stress. So this is about 32 to 34 Celsius degrees. Right? So you can see the movements. So they have very kind of huge movements. So these are called flank movements. So we are kind of counting that flank movements all the time, every hour, every minute, every 2 hours time. So we just try to count them. And if we feel they are going above the standard levels, we just turn off the heaters and just try to cool them down. Right? So this is how they are kind of trying to deal with the kind of a heat stress or the heat stress. So we have to observe. Observe stress level. Infrared. So we just kind of scan them whether the blood flow is to the outside of their body, right? Otherwise, they are just like this. They are always kind of closer to the water and just try to drink. Drink bonnet and just kind of stay close to the waters all the time. But they always try to kind of cool down their body. So always close to the waters and urinate more. That's the main thing. Right? So all 12 hours they are just like this. So once we turn on the heaters, so they always kind of moving their body all the time. Because their body surface area to body mass ratio is very low. But if you compare with the mature ones, so they are even heat stress. අපි ගොඩක් වෙලා ලොකු සත්තු ගත්ත මීට වඩා ඒගොල්ලන්ට අමාරු වෙනවා ගොඩක් වෙලා නිකන් වැල්ලා ඔය ඉන්නවා නිකන් මැරලා වගේ අපි දැකලා තියෙන සමහර ලාවට මේ නමුත් ඒගොල්ලෝ එහෙම නැහැ නම් the heat stress, that's how they are trying to deal with whatever the stress they may have, right? So then we did several calculations with the models just to kind of calculate whatever the energy partition, so what could happen, whatever the things that they consume, right? Because they are not growing. So I will show you some kind of results. So this is just to show you the heat level. We usually measure them through the temperature humidity index. So for the dairy calves, which is dairy cows, usually the mature one, which is about 65 to 70. අපි සාමාන්‍යයෙන් mature cow කෙනෙක් ගත්තොත් pH index කියලා අපි කියන්නේ ඒක සාමාන්‍යයෙන් 65 70 වගේ යනකොට ඒ සත්තු ස්ට්‍රෙස් වෙනවා. කෑම කන එක නවතිනවා, දෙන එක අඩු වෙනවා. That's what happens for these animals. But the young ones, they can tolerate a little bit more than the mature ones. So they usually kind of can tolerate maybe about 75 to 80 that pHI level, but during our research they were under about 85 or even above 85. So then we can kind of confirm that they are basically heat stress conditions. So therefore kind of a successful kind of a initiation of giving some heat stress for these animals. So otherwise we cannot say they were kind of under stress. So other measurements were just to kind of, we were measuring the rectal temperature, skin temperature, and the heart rate and the respiratory rate. So when we compare all these values with the baseline values, we saw kind of a significant increase in the heart rate, rectal temperatures, as well as respiratory rate and the skin temperature. So which confirmed that, so they were under heat stress. So that's what we wanted to do actually. අපි ඕනේ අතරම ඒගොල්ලෝ mimic කරන්න. මේ condition එකට මොකද වෙන්නේ කියලා. Because in Iowa, where I was doing my PhD, so during the summertime, the temperature is kind of going up even about 35 in some cases and lasts for maybe about 7 days at least. So which means it's called a kind of a heat wave. ඒගොල්ලන් heat wave එකක් කියලා කියන්නේ සතියක් විතර එක දිගට එක විදිහටම තියෙනවා. උදේ ගත්තොත් උදේ 6 විතර පටන් ගන්න 6ට එළියට බහින්න බෑ. ඒ තරම් දාඩි දානවා. කියන්නේ දාඩි දාන්නේ නැහැ sorry. We do not sweat there because the RH as well as the temperature. So these animals cannot bear that kind of a situation. මොකද අයවල ගොඩක් තියෙන issue එකක් තමයි RH එකත් වැඩි, ඒකම temperature එකත් වැඩි. So therefore we cannot sweat. So therefore we had that kind of issue. So that's what we were trying to do even during this research. So we were kind of successful in that regard, but however, we did not see kind of a significant effect on phytogenic feed additive supplementation on these particular parameters. අපි ඒ වුණාට දැක්කේ නැහැ විශේෂයෙන් මේ parameters වලට ලොකු බලපෑමක් වෙනවා අපි දෙන phytogenic feed additive එකේ. Maybe because of the heat stress is not enough for these animals, or maybe we don't know. Maybe any other kind of a effect had some kind of an issue just to kind of avoid or kind of a stop any effect on these phytogenic feed additives. However, if you kind of see the dry matter intake, we saw some kind of an effect. Increase of heat, decrease the dry matter intake. අපිත් එහෙම අපේ product වලට මිනිස්සු වුණා heat එක වැඩි වෙන්න වැඩි වෙන්න අපි කන ප්‍රමාණය ගොඩක් වෙලාවට අඩු කරනවා, because if you eat more, what happens is digestion process will create lots of heat, which means it will double the amount of heat load in our body. So if you cannot sweat or kind of dissipate whatever the heat, so which means you are feeling very uncomfortable. So that's what happens to these animals as well. So they have kind of a method just to kind of stop or kind of reduce the amount of feed that they consume during the stress period. And once the stress is kind of released, they just start to eat a lot of food. So that's what we saw. Even with this phytogenic feed supplementation, what we saw was they try to eat some more about feed compared to the control group. Control එකට සාපේක්ෂව මේගොල්ලෝ ටිකක් වැඩිපුර කන්න ගත්තා phytogenic feed additive එකේ බලපෑම නිසා කියලා අපි ඒ වෙලාවට suspect කරනවා. But we cannot certainly say why, but based on these two kind of observations, that's what we believe. Okay, the supplementation of this additive kind of enhance their feed intake to some extent. Then we check the blood gases as well, the pH, carbon dioxide, the potential, the partial pressures, the excess gases, sodium, potassium levels, and glucose, hemocritic, and the hemoglobin levels. So we saw some kind of increase in blood oxygen concentration and hemoglobin levels in these animals. So it's kind of a suggestion that the phytogenic feed additive kind of might have enhanced the blood flow towards the lung and enhance the kind of oxygen intake through the lungs of these animals. Because the main claim given by the company regarding this particular product was whatever the blood will be kind of directed to the periphery of the body. මේකල ගොඩක් වෙලාට කිව්වේ මේ product එකෙන් කරන්නේ heat එක dissipate කරන්න facilitate කරන්න ඒ තියෙන what you call the blood flow will be directed toward the skin. So that's what they were kind of claiming. So we also kind of had some evidence, but not 100% true, but still we can say there was some blood flow kind of a direction towards the lungs as well as for the skin, just to kind of enhance the heat dissipation through the skin of these animals. So then we were testing some inflammatory markers through the ELISA. So we tested the haptoglobin levels and LBP levels. This is called the inflammatory markers. Those are the basic markers that we usually check whether the animal is in sick or not. Animal sick නම් නැත්නම් sick වෙලාද නැත්නම් එහෙම නැති වෙලාද කියලා check කරන්න අපි use කරන ප්‍රධානම ELISA දෙක තමයි මේ තියෙන්නේ. So haptoglobin උයි LBP තමයි අපි ගොඩක් වෙලාවට check කරන්නේ. LBP means lipoprotein binding proteins. So those things were tested. So we saw some kind of a significant effect on supplementation of phytogenic feed additive. So which means the supplementation of that product decrease the haptoglobin levels as well as the LBP levels of these animals. So which kind of a suggestion they are supporting to kind of decrease the inflammation of these animals under stress conditions. Then we tested cortisol level just to kind of see any kind of stress levels, but we did not see any effect on phytogenic feed additive supplementation of these animals. And lastly, so we had some oxidative stress markers, lactate, glutathione, T-bars, and carbonyl content test. So all were kind of supporting to some extent the phytogenic feed additive supplementation had some positive effect on these animals under heat stress conditions. So finally, as conclusions, so we had some basic conclusions under this particular research through the field research. So the animals, yes, of course, they were heat stressed because the temperature was very high. They above 40 in some cases, in some times. Because අපි ගත්තේ ගොඩක් වෙලාවට පැයෙන් පැයට ගත්ත temperature ඒ ගන්න වෙලාවට සමහරවිට සෙල්සියස් 40 පැනක වෙලාවලු තිබ්බ මේ සත්තුන්ගේ ඇඟි. ඒතර ගොඩක් ඒ වෙලාවට තමයි සත්වර කරගන්න දෙයක් නැතුව වැහිලා ඔහේ ඉන්නේ. නමුත් මැරුණේම නැහැ. but තව එකක් දෙකක් උඩට ගියා නම් ඉවරයි. Right? So then the dry matter intake just went down to some extent, and supplementation of this feed additive just enhance dry matter intake in some animals and some conditions, and also kind of inflammatory markers. So they were kind of having low inflammation under these heat stress condition when supply these kind of phytogenic feed additive. So that's the basic things actually I wanted to kind of highlight today, and I would like to acknowledge all the members. So I had a 5-member PhD committee over there. So they were the ones kind of supporting me, and all the other members and the other universities surrounding in the USA. So they were basically supporting me to just to kind of obtain some support their lab research, and the USDA, specifically United States Department of Agriculture, they were helping me a lot. And also our university, the former head of the departments, former deans, and the vice chancellors helped me just to kind of approving my progress reports and providing whatever the leave without any kind of a restriction. So I was able to finish this one with the given time period. And special thanks goes to these animals. So they were kind of with me about 5 years without any kind of issues, because I was restricting their feed, water, taking blood. I don't know, I did almost all the kind of troubles to these animals most of the time. So, but they were helping me a lot just to kind of support to kind of finish this one. However, there were some animals, they had to sacrifice their lives as well, just to support my research. So that's the basic sad thing I have, because I was raising them since the day they were born, and maybe after some time, so I had to sacrifice these animals just to get some tissue samples, blood samples, and those kind of things. So that's the only regret I have selecting animal science and doing animal science research. But I think they sacrificed their lives for the betterment of their colleagues or whatever, friends or whoever, their industry in the world. So hopefully the results that I kind of got may have some effect on the development of dairy industry. But yeah, because the same animal, the same image from the same, this is the day before this one is going to die, and this is the day of I took the samples. So මේගොල්ලෝ මාත් එක්ක 31 දවස් ඉස්සෙල්ලාම හිටියා. ඊට පස්සේ ඉතින් කරන්න දෙයක් නැහැ. So in recognition, just to kind of additional information, at the end of the PhD period, I was able to kind of obtain this award. This is called the Research Excellence Award offered by the Graduate College of Iowa State University for the research did for my PhD from the animal science degree. So for that year, I was the recipient of this research from the university for the animal science department. So all the sacrifice, everything kind of paid off ultimately, but with the sadness of these incidents, but still I am happy I was able to get the Research Excellence Award from the university. And after completion of my PhD, so I was able to publish almost all the chapters of my PhD thesis. There were 6 first authors referred peer-reviewed journal articles, 3 industry reports, and about 9 or 10 peer-reviewed abstract already published. Only literature review is to be published. Hopefully, it will be also published pretty soon. And then, yeah, it's kind of a journey, but both happy and the sadness. But fortunately, I was able to kind of finish almost all the things amidst COVID. The research that I was showing you, which was conducted during the peak time of the COVID, therefore there were no any support from the university, because they were asked us to stop all the research, but unfortunately we already started the research, so then we could not stop that one. So even the professors, all the committee members were there. Sometimes they came and helped me, and even as an international student, I was kind of lucky to get the support from these professors, because Iowa State means kind of a state which is called as red state. So you know the red state and the blue states means, so which means they are not more helpful for some people, but still as an international student with this kind of a skill, they were helping me a lot. And ultimately I was able to kind of achieve this kind of a target, and within the given time period. Only 2 months were kind of delayed because of the COVID. Otherwise, within 5 years of time, I was able to finish almost all the things. So that's what I want to kind of present you today. So these were the animals. So these two are no more with us, already died. This one is hopefully still be there. So මේගොල්ලන්ට තව අර මම ගත්ත කිව්වා රිසර්ච් එකකට ගත්තා කියලා මේගොල්ලෝ මාස 3කට පස්සේ අපිට සැක්‍රිෆයිස් කරන්න වුණා ඇනිමල්ස් 40ක් මේ ඒ රිසර්ච් එක ඉවර වුණාට පස්සේ. So ඒගොල්ලන්ගේ tissue samples තාම මම හිතා lab එකේ ඇති, but we did all the analysis and we already published whatever the information. But we had to sacrifice these animals, but hopefully she is there if nothing happened with any other kind of research group. So that's all what I have to say. So if you have any questions, I am happy to answer. But this was kind of a sudden preparation, so that's why I am not kind of maybe missing some information, but I feel some kind of feelings when I see these images, but I wanted to share that with you as well. Thank you.
Okay. Thank you very much, Janaka, for your wonderful presentation, and I think there are a lot of questions from the audience. I will ask one. Maybe I'm new to this kind of research. I have not done this kind of research. But when you measure things in animals and say that these have an effect or not, how do you have the control? What is the control for the animal? Is it before and after in the same animal, or you have other animals as control?
We have a separate group.
As control?
Yes, we have for this one, the previous research, we had one group as the control one, so the other one as the treatment group. So only.
But then how do you take the impact of the animal effect? Maybe some animals are more vigorous or healthy.
Yeah.
Some animals may be not that much.
Yeah. It's basically randomization. Initially, we randomly allocate them based on the body weights and whatever all the parities and all the kind of parameters. But as you said, yes, that effect is there. But almost during the model, statistical model, we as much as try to kind of avoid those things. But if we see any extreme cases, we just take them out.
Is there any, just for my knowledge, is there any way that we can use the same animal as the control and the treatment? No?
Yes, we can do that. Latin square designs in some cases, but these ones are not cannulated. So we have to go with that kind of a thing. So otherwise, the main thing is the funding agencies. They are usually funding for a particular time period. So if you are using the same animal, which means it take a longer time. So which means everything they are considering the money most of the time. So then we cannot use the same animal in some cases, but for the mature ones, of course, the cannulation and everything, Latin square designs, yes, we can do the same animal after washout period for the same research, same group, so we can prospect, no issues. But for these animals, no, because they usually have some demands. Okay, we have these animals, we should have, at the same time, we have to conduct the same thing and get the results at the end of the day.
All these animals are they for milking or are they for
milking? Means these are young ones, which means the dairy Persian cattle, the young ones, ladies.
What I'm asking is this not for meat, right? Meat is
dairy. This is dairy cattle. Yes, these are dairy. Or sometimes these are pure Persian actually. Pure Persian cattle are made.
Persian are not the meat told. Dairy period once they
are done, they just go to the market.
But for specifically animals who are they found specifically for meat?
Yes.
Beef
cattle industry, yes.
What kind of animals are they? Are they Frisian or?
No, they are called Angus. Angus. They are the main popular breed is Angus. But these ones also, once they are done with their production and all things, they just go to the same pathway as the Angus.
Okay.
Buy at the market.
Yes, well done, Dr. Janaka. I think it's a real life experience in research, right? You have done a lot of
So even you can see this one. Some research were conducted even during the winter season,
Yes,
which
yes.
means the outside temperature is minus 30 Celsius degrees most of the time. So we had to be outside almost all the day. Sometimes bleeding is not really, all the hands are freeze, all the frozen, but we had to do that.
So I think it's a good. You have shown some good research. You are good. So I wish you can do some contribution in Sri Lanka also to identify this. You said the signs for the stresses. In the industry.
That's what I am actually trying to do these days. But the thing is, the facilities and the resources available is very minimum. But still trying to do some kind of a contribution.
Just a clarity now. You check the signs. How about the milk yield? Got a time to check that?
No, these are actually the young ones. Maybe maximum 0 day to maximum we go maybe about 16 to 20 weeks. So we had some kind of a data because.
So what is the economically benefit? There is for that cost reduction.
Cost reduction. Yes. සාමාන්‍ය එක සතෙක් සති 9ක් අදිනවා නම් ඒ කාලේ 2019 21 කාලේ ඩොලර් 380ක් විතර එක සතෙකුට වැය වෙනවා ඒ කාල සීමාවට. අපිට ඒක අඩු කරන්න පුළුවන් සතියකින් නැත්නම් දෙකකින් ඩොලර් 50කට අඩු කර ගන්න පුළුවන් එක සතෙකුගේ වියදම ඒ කාල සීමාවට. ඒතකොට සත්තු 2000 3000 කිනක අපි දැන් අපේ ෆාම් වල එහෙම නෑ. ඒ වගේ රටක සාමාන්‍ය ඇවරේජ් ඉන්නේ 3000 5000 වගේ අතරට. ඒ වගේ රේන්ජ් එක හිතලා බැලුවොත් ලොකු කොස්ට් එකක්. Labor cost, everything.
Ultimate quality of the product is?
There is no difference.
No difference.
Even we checked, even during my master's I did preparatory research, so we were testing a similar kind of a thing. So we checked after 5 months of age and even during the first milking time, so we did not see any difference. So
I had a funny question. You said there were urine sample. How much urine ekatu kore?
ගෑනු සත්තුන්ට කැටි එකක් නෑ කැටි එකක් දාන්න පුළුවන්. කැටි එකක් දාන්න. ගෑනු සත්තුන්ට ඒක අමාරුයි කරන්න.
Watching all the time I thought.
ඒක මම පෙන්වන්න අපිට වෙන මෙටබොලික් බාන් කියලා එකක් තියෙනවා. මෙටබොලික් බාන් එක දාන්න පුළුවන් මේ සත්තුන්ගේ යූරින් කලෙක්ට් කරන්න.
Any other question? නවෙන්ට ප්‍රශ්නේ දෙන්න. Now, you did 4 small animals, right?
Yes.
Is there, now you are talking about stresses, how to minimize the effect. Anything that has been done for the milking animals, or is there yield reduction? Because
yes,
if I did this, I would go for, I don't know, I would go for a milking animals to understand is there any yield reduction. That is what we usually do. We are going for the younger ones. So what would be the
yes, actually lots of things have been done regarding the mature ones. The thing is, these are the replacement stock. So we wanted to see whether any effect on the similar kind of stresses during their young age would have effect on whatever the future kind of performances and those things. So the lab is kind of investigating the future result because it will take even another 1.5 years just to start giving
continuing into
they are continuing with the research, but this is the kind of initial part of the research. මුලදී අපි කරේ මේක දැන් බලලා මේ කාලේ වෙන ඉෆෙක්ට් එක මොකක්ද කියලා බලනවා. ওই සත්තු ටිකම සිලෙක්ට් කරලා සමාන සත්තු ඉතින් මගදී මැරෙනවා. ඒක හේතු නිසා කල් කරනවා අයින් කරලා දානවා. නමුත් මැජොරිටි කොහොමද සර්වයිව් වෙනවා. ඒගොල්ලෝ කොහොමද ඊළඟට වෙන්නේ කියන එක තමයි ෆියුචර් රිසර්ච් එකේ විතර ඒකට කන්ටිනියුෂන් එක විතර යන්නේ. ලැබ් එකේ. අපි අපේ ලැබ් එකෙන් සමහර ඒවා කරන්න ලැබ්ස් කීපයක් එකතු වෙලා ඕවරෝල් කරගෙන යනවා සමහර ඒවා. ඩේටා කලෙක්ෂන්ස් ඔක්කොම කරනවා. මොකද අපිට ඒගොල්ලන්ගේ ඩේටා ගන්න පුළුවන් මේ ඒ අවශ්‍ය වෙලාවට ඒගොල්ලෝ ඔෆර් මේ කිසි මේකක් විතර ඔෆර් කරනවා. දැන් මෙතන මැචුවර් එක කරපු එකක් මම හිතන්නේ මම හරියට එක්ස්ප්ලেইন කරගෙන නැත්නම් ඉස්සෙල්ලා මේ ඒක අපිට ඩේටා එකක් තමයි සාමාන්‍යයෙන් දැන් මැචුවර් කරලා තියෙනවා ඒගොල්ලෝ වාගෙන තියෙනවා PHI index එක 65 70 වගේ වෙද්දී ඒගොල්ලෝගේ intake එක ටක් ගාලා කඩාගෙන යනවා. ඒ කඩාගෙන වැටුණා කියන්නේ so what happens eventually their milk production is also going down. Drastic reduction, maybe about 30 to 50% reduction can be seen because of the stress. So they are losing if I am correct, the estimation is about 1.5 billion of US dollars for one particular year. They are losing money because of the heat stress. 1.5 billion US dollars per year. That's the average. I think it's higher now compared to the previous years. It's kind of a big research area. So different universities, different departments, research group, they are kind of doing separate research and eventually coming into one kind of a findings at the end of the day.
Any other questions? කාටරි ප්‍රශ්න තියෙනවද? නෑ ළමයි වැඩක් නෑ.
තේරෙන්නේ නැතුව
හරක් වරියයි කියලා තේරෙනවා.
It's also kind of we were testing a product. So we were testing a product, so that's why we just sacrificed them. So we had to take some samples, liver samples, digestive systems and everything. We had to collect even heart, skin, everything, fat. So we wanted to kind of measure the residue level. So that's why we had to sacrifice at the end of the day those animals at the end of the period. So that's what we did.
Yeah, Dr. Janaka, very interesting research. And how can you apply your findings under Sri Lankan context, though we don't have a large industry?
Yes.
At a small level, because particularly in the dry zone area?
Yeah, so they are basically having the same effect actually. So one of the reasons, even I talked with Professor Cyril, he is the chairman of NLDB. So that's the main thing, because of the heat stress. So they are losing lots of money. So maybe they do not have resources, so they are not kind of initiating those things. And even as I mentioned initially, they are waiting until 4 to 6 months to cut down the milk. So they can save lots of money actually, because they are giving about 3 liters for 6 months. So just imagine the amount of volume of milk that they are offering. So if we can at least cut down maybe 1 week, 2 weeks, we can save lots of money. So, but the thing is, I don't know, the farm managers, or they are not accepting that, but because they want to kind of practice whatever they have been practicing maybe many years. So that's why even in USA they are not accepting most of the things, because they want kind of a research, fine research, so then they will accept. But the thing is, it is very difficult to kind of convince them, because they have their traditional experiences. They are arguing and they are not kind of accepting, but at least they are now going for at least 6 weeks. 6 to 8 weeks, but our country we are still going for 4 to 6 months.
Is that with the Batu Harak or other?
Even with the other ones, the Indian cattle breeds, the Sahiwal, Tarpakar, so even the crossbred, even for the other ones, even we went Ambevela farms very recently for a field visit, even still they are feeding milk until 4 months. So I asked why you have feeding 4 months, because they are usually becoming a ruminant after 6 to 7 weeks. No answer, but that's what they are practicing. So that's the thing. So they might be thinking other way, but it's waste of money actually. So then at least we can increase to some extent our milk production, at least 1-2%, which means a huge volume. They are feeding about 3 to 4 liters per day per one cow, which means if you multiply by that volume with the total number of dairy calves we have, which is a huge volume of milk.
So can you give a sort of recommendation to the NLDB? So
I already talked with Professor Cyril. I don't know whether it will be kind of accepted or not, but that's what I am trying to do, some research with NLDB. So then some kind of research with other students, even we did one research last time as well. So at least just to show them, at least farm wise, just kind of accept this one. Hopefully they will accept someday, but you never know. So that's the thing. So it's very expensive. Even 1 liter of milk is about 200 rupees. So we can save it, plus labor cost and everything, we can cut it down.
So first of all, thank you very much for your insightful research. We were really inspired by that. So my question is, in Sri Lankan context, we know that most of the research findings do not go to the policy implementation levels, but I can see your university also in state university. So what I want to know is this quite a general question. What I want to know is, since there are very conclusive findings and findings are really very relevant to the industry, was there any signs of these going to the policy implication levels?
Yeah, actually in USA university system, they have in the faculty, in the department, which is I was in animal science department, they have a separate faculty members called extension faculty members. So they are only responsible for extension services. So what they usually do is the findings of the research faculty are disseminated through those faculty members to the industry or the farmers or whoever, because they are given a kind of a targeted industry or the farms in the state. State means it is almost 2 times of the size of our country, which means it's a huge state. So huge means comparatively, but if you go to any other state, which means 10, 15 times than our country, but Iowa is also kind of a 2 times the size of the Sri Lanka. So if you kind of see, they have a separate faculty members, a group, extension group. So those are also professors, senior professors. So they are the one responsible for this kind of a task, just go and doing the dissemination. So they have research faculty separately, teaching faculty separately, and extension faculty separately. ඒගොල්ලන්ගේ එහෙම setup එකක් තියෙනවා. දෙකම තුනම කරනවා තියෙනවා. අතරින් පතර. නමුත් ප්‍රධාන වශයෙන් faculty 3කට විතර කඩලා තියෙන්නේ. ගන්නම extension faculty කියලා. ඒගොල්ලන්ට උගන්වන්න මුකුත් කරන්නේ නැහැ. තියෙන එකම රාජකාරිය තමයි අර ඔක්කොම findings ඔක්කොම ගිහිල්ලා industry එකත් එක්ක university link කරන එකයි, dissemination කරන එකයි, knowledge sharing කරන එක. ඒක තමයි ඒගොල්ලන්ගේ main task එක වෙලා තියෙන්නේ. research කරනවා research විතරයි. ඒගොල්ලෝ research වලට විතරයි responsible, teaching මොකුත් කරන්නේ නැහැ. teaching faculty teaching විතරයි. ඊට අමතරව සමහර faculties ඉන්නවා යම් ratio එකක 60% research, 40% teaching, එහෙම නැත්නම් 60% research, 20% teaching, 20% extension or something like that. So they are responsible for almost all kind of things. That's how they are kind of have a setup, but different universities have different setup, but in general that's the basic setup over there. So there are some faculty members, they are responsible for dissemination these findings to the industry or the farmers who are belong to the Iowa State University or the kind of a boundary of the Iowa State University. So that's what they are doing.
So there is another question. What is the optimum temperature range for the Friesian breed that which they produce the most quality milk and the amount wise?
15 to 25 Celsius. That's optimum 15, but still they can bear maybe about 25 Celsius degrees. But the THI levels will go up if we kind of increase the temperature, which means their milk will go down. So, but on average 15, that's the optimum. බින්දුවේ ඉඳලා 15 වෙනකන් රේන්ජ් කරන්න පුළුවන්. අපිට නුවර ඉලෙවල් කන්ඩිෂන් දෙන්න පුළුවන් නම් හොඳටම හොඳයි. නමුත් ඊට වඩා වැඩි වෙන්න සෙල්සියස් අතක එකක් වැඩි වෙන්න ඊට ප්‍රොපෝෂනේට්ලි අනිත් පැත්තෙන් අඩු වෙන්න පටන් ගන්නවා. That's what happens.
Okay, thank you very much. Thank you very much, Dr. Janaka Wickramasinghe, for the wonderful talk that we had today. And it's a wonderful event and very, very informative. And we learned a lot of things and from animal science research. And this is what why I have asked everyone to come. I don't know how others, why others didn't come, but let's start this, let's continue this, making all students to attend this, somehow all students to attend this. The reason is that we get more ideas when we see other things, what other departments, other researchers do than what we do. And there was a nice answer for the question raised by a student that the setup in the US university system, they have a separate unit to disseminate information. Maybe that is where we appointed our Sagar today for the external part. So we do only kind of teacher training and all that, connecting with the industry, but we can go further, like Dr. Janaka said. So we will try to go in that path, but unfortunately we have only very few staff members in the faculty. So that is why we are very unfortunate. So therefore even his finding can be applied in the Sri Lankan context, as we understood, as he has explained. So thank you again for giving us a wonderful talk, and we will meet again in next month, first Wednesday, with another wonderful talk. Right? Thank you so much for participating.
Thank you very much and for the given opportunity, sir. And yes, I think you got something, but hopefully we can have some other kind of a discussion later as well if you need. So thank you very much and have a good day. Thank you.